Let's take the last session before the lunch break, and this is on another important aspect, which is GMP compliance. And we have as a session lead, Dr. Giampiero Lorenti. He's a senior GMP inspector at the Italian Medicines Agency Inspectorate. He's also a member of the GMP GDP Inspector Working Group at the EMA, and also a member of the PAT expert group. So please, Dr. Lorenti. Thank you. Thanks very much.
Thank you very much. The next slide, please. So my presentation will be a part of a general overview, very short overview on GMP, importance of GMP. It will deal with GMP topics that were raised during the evaluation prime application in Europe. Next, please. Why the GMP are so important? The GMP is important because it's a part of the pharmaceutical quality system that ensures that medicinal products are consistently produced and controlled according to the quality standard they are appropriate to their intended use, and also as required to the market authorization and the clinical trial authorization. And also it's ensured that the medicinal products that are placed on the market do not place patients at risk due to inadequate quality and then safety and efficacy. It's an important part of the prime because it can ensure that the product released no any problem on quality, so will not impact on the prime process, in the prime process itself. The next slide, please. This is a general review of the legal framework in Europe. On one side, we have the legislative framework made of regulation and directive that lay down the related provisions. And on the other side, there are the guidelines, the GMP, that is composed of chapters and related annex that provide clarification of specific types or product. One important thing of the GMP that the GMP indicate the point to arrive, know how to arrive there. This is important to take into account. It's important that in the last few years we have a global harmonization of GMP, also thanks to the MRAs and the PICs, but in particular to MRAs that provide the possibility to harmonize system between different regions. Next, please. The first topic that I would like to see illustrate is a submission of conditional marketing authorization application based on clinical trials data generated in non-GMP site. So this was addressed during prime application, and as you know, in Europe, the manufacturer investigation of medicinal product or TMPs should be in compliance with GMP. And in case of products that are imported from a third country, the importer shall ensure that the products are being manufactured in accordance with standards that are equivalent to the EU GMP. And the application of GMP is important, as I said before, because it ensures that the subject of the clinical trial are not placed at risk, and also that the results of clinical trials are not affected by inadequate quality arising from unsatisfactory manufacturing. Next slide, please. So during the prime application, it was asked to provide an evaluation of gaps analysis, considering that the site was not GMP because it was an academic laboratory. And here I have listed some points that were part of this evaluation gap analysis, and I've highlighted in blue. I don't see if it's blue. Probably okay. So, but okay, in the original slide there was blue about some points that indicate a failure regarding the samples that we have seen. For example, there was no evidence of PQS. There was no possibility to perform an inspection later. So, in order to assess if the site could be considered compliant because it was closed. And there was no system in place for handling OS or T, so they were recorded but not a full assessment was done. Also the change system was in the same condition, documentation handling was the same, so batch record, there was no traceability of what happens during the manufacturing, so it was more possible to evaluate clinical batches and comparability data between the different manufacturing process versions. Next slide, please. So another point was concurrent validation. We have already discussed about it. As you know, in GMP, it's written that the product that should be released and the process is the product should release it to the market should be validated, and respect to the prospective validation, there is also a possibility to have a concurrent validation, but this should be acceptable in acceptance, should be acceptable in exceptional circumstances. So when, for example, there is a strong benefit-risk ratio for the patient, or in case for the ATMPs, in case of limited availability of the starting material. It's important that the regulatory authority should be involved in the decision and also in the strategy. All the process should be justified, all the decisions should be justified, documented, and approved by the authorized person. Part of the possibility to accept a concurrent validation is to have to design and implement a robust validation approach. That it means robust validation strategy, protocol, correct definition of the control quality attributes of control process or CPP, and associated acceptance criteria based on the development data and process knowledge. An overview of all the production process version used by using also data coming from non-clinical and clinical in order to achieve the confirmation that the manufacturing process is able to ensure consistent product quality and the defined specification are comply with. The next slide, please. Auto spec. This is another point that was raised. So, as you know, in GMP, the auto spec is part of the inspection activity, and any auto spec should be fully recorded, investigated with the objective to determine the correct root causes possible, and implemented obviously appropriate corrective and preventive action. Part of the process of the investigation is also to implement a quality risk management process. This is important in order to achieve the result we would like to see. And during the OS investigation, the qualified person should also consider to include the affected batch instability and possibility and potential for batch variation in case of recurrent OS. It's possible in exceptional case that a company should have to release a product on the market in case, in spite of an initial OS that has not been validated. But this should only come after a full investigation has shown that the OS does not reflect or impact quality, safety, and efficacy on the batch. And in making this decision, the QA and the QP should always be on the side of caution and strongly justify the decision. This is in particular, next slide, please. This is already foreseen in the ATMP, so in the new part 4 that was drafting for dealing with the ATMP. And I listed the points because it's clear from when we discussed the part 4, it was clear that there will be the possibility that the need to release product with OS on the market. Also in this case, obviously, exceptionally, it could be evaluate the possibility to release in order to avoid obviously significant hazard, take into account alternative options, but also considering the consequence of not releasing the product for the patient that could die if he will not receive the product. So in that case, it could be justified to release and to provide, to supply the product to the treating physician. What is important is to have a continuous sharing of information between manufacturers, the treating physician, the marketing authorization holder or the sponsor, and the regulatory authority. So in order to exchange all the time information about the possible risk and everything that could impact. Another point that I already mentioned is that in case of recurrent deviation, the need of change for manufacturing process should be assessed, but also for variation. Next slide. Master cell bank or working cell bank not manufactured under GMP. So, as you know, the establishment of master cell banks should be done under GMP. The use of no GMP bank can be only accepted in exceptional case and provided that there was an extensive characterization and testing. That it was done also by including a deep risk analysis or deep risk management process. And in order to really understand the testing requirements necessary to ensure the quality of the starting material. It's important that in this process, the competent authority should agree about the strategy. The next slide, please. The last point is about batch release from laboratory based on third country. And as you know, according to our directive and as reported in the Annex 16, the QP is responsible for ensuring that each product, manufactured product, have been manufactured in accordance to GMP. Unless there is a mutual recognition agreement or similar agreement, the QP is also responsible for ensuring that all the lots were tested for a full quantitative analysis and a quantitative analysis of all active substance. There is the possibility, in exceptional case, to skip this test. This is mentioned in part 4, but obviously it should be justified. It should be justified, for example, in case where there is a limited amount, for the part 4, a limited amount of material available, or in case of short shelf life. I mentioned USA in the title because there is a misunderstanding regarding the mutual recognition agreement with the USA. The fact that we started with the mutual recognition agreement, it doesn't mean that at the moment in EU it could be not performed the test. So this will be skipped only at the end of the system, when all the member states will be recognized. So in 2019. The next slide, please. I would like to thank the people from EMEA and Italy that has helped me and support me in this presentation. Thanks. So I think that we can move to the presentation of the case study. I would like to introduce Matt Popkins, that is from GSK and co-lead of FPA EBE CMC Accelerator Access Team. Thanks, Matt.
Thank you, Giampiero. And just want to take a moment on behalf of the FPA EBE, Vaccines Europe, Pharma and Bio group that we convened to support this workshop, just to thank the EMA and the FDA for the discussions today, already been really rich and hoping for some really fruitful outcomes. And just to turn it around, to acknowledge all the colleagues who worked with me and my colleagues Marcus on the industry group, who put an extraordinary amount of effort into this workshop. So thank you, everybody. So if you carry on, please. It's obviously a very broad topic, GMP, so we're going to limit ourselves to talking about a few specific topics. And I'll just go straight into it if you go to the next slide. So the first slide I've got is really articulating the foundational principles behind why we do GMP and the points that Giampiero made about the fact that GMP is already well harmonized globally. What we want to focus on, obviously, today is the considerations for accelerated asset. And the most important point in all of that is the first point we've got there, which is that obviously continuity of supply to patients is obviously the main consideration we need to think about. And I'll harken back to the points made in the case study on validation that Stuart talked about. That's just an opportunity for me to say we're not going to focus on validation, particularly in this session, because we had a good discussion on that earlier on. So I won't carry on with this slide. I think it might be easy if I go to the next slide, which I'm going to try and use an image just to sort of articulate a point. So in a traditional approach to development, we usually have a relatively long clinical investigation, phase 1, phase 2, phase 3, before we move into commercial phase. And obviously our quality, CMC understanding, our processes, our control strategy, etc., builds as we go through that, and at some point, typically late in development, sometime in phase 3, we're developing the final quality or CMC package that supports the commercial product. Now, the same is true in the GMP space. We're using often different facilities that are used to make clinical or IMP material in the clinical phases, and at some point will transfer into the commercial manufacturing facilities. And a lot of what we've been talking about today, and we'll continue to talk about, is taking a different approach to the quality and CMC elements that support accelerated access. So at some point in the late clinical phase, in the early commercial phase, we're doing something different from what we've been doing traditionally. And so the question we're asking is, does that also apply in the GMP space? And that's what we'll explore briefly in a moment. So there's just two bills if you click on that and click again. So the key consideration in the accelerated access scenarios is that the concept of commercial GMP considerations is related to GMP standards for the whole of the commercial life cycle, whereas what we're talking about here is supply to patients who are critically ill for a relatively limited period of time. And so we're talking about that scenario and that scenario alone. Okay, if you carry on. Okay, so the case study or the example scenario that we've put together from FPA companies is actually one that we think applies relatively frequently already in accelerated scenarios. So, a cancer product where the early clinical data shows efficacy, and where phases 2 and 3 are therefore a single study which is supplied from a clinical IMP site. So obviously here, as we said already, the imperative here is rapid file and launch in ensuring continuity of supply. In a lot of cases, it's possible to determine scientifically that the introduction of the commercial manufacturing site, which may be a relatively small, if any, increase in scale, very similar manufacturing equipment, that is often on the critical path, but actually scientifically and technically, it can be relatively straightforward to justify or determine that the product will be equivalent. But sponsors need to use the commercial facilities to supply the commercial product, and don't believe that the commercial supply can come from an IMP site, with the obvious example of the AstraZeneca validation proposal this morning being an outlier in that space. So thinking about clinical supply sites, there's a lot of good reasons why clinical supply sites would be good for rapid commercialization and why they're appropriate in accelerated scenarios where there's less knowledge and less experience, less understanding. And I won't list them all out, but it's an obvious point that they are skilled at rapid introduction of new products. They are skilled at learning from scale-up and from experience, and they have systems and processes and expertise that are readily adapted to those. So we're just going to take a few. There's 4 points that stem from this scenario that the group put together to pose for the discussion. So if you carry on. The first of all is overall GMP considerations. So if GMP considerations for the clinical phase are appropriate for the clinical phase, are they also appropriate for early commercial supply where there's unmet medical need? So there are some clear differences between clinical expectations for GMP and commercial expectations for GMP, but actually there's the foundation elements are obviously very similar, but equally maybe there is an opportunity to look at the overall expectations for clinical and commercial GMP and consider actually where the essential points for commercial supply can be met from IMP sites. So if you carry on. So in terms of commercial supply from IMP sites, so we've talked about the scenario, which we think is a common scenario where a sponsor, an applicant may choose to use those. And we talked about the fact that scientifically it can be readily achievable to demonstrate and justify equivalence of the material from an IMP site to the commercial product. But even within IMP site, product licenses differ across EU member states and across regions. But the most fundamental point is that in every region, there's a requirement for a commercial GMP license in order to supply a commercial product. So the question we're asking is it possible to supply commercial product for a time in the early commercial phase from an IMP site which doesn't have a full GMP commercial license? So if you carry on. So related to all of this is an obvious point, and we've heard a lot about this already today, and we'll hear more, I'm sure, around the importance of dialogue with regulators through scientific advice and other mechanisms. So it's clearly a fundamental point to progression of prime and breakthrough therapies, and is obviously incredibly useful in determining what is an appropriate control strategy and data package, et cetera, et cetera. The question is, are there equally robust mechanisms to ensure that GMP considerations can be discussed? So with inspectors from OPQ as part of those types of meetings, or perhaps with inspectors working group members as part of scientific advice discussions in the EMA. And again, I won't list through the topics to agree and it could be useful to discuss in those types of meetings. But it's clear that what industry needs, and I think again Stuart referred to this point earlier on, is certainty that outcomes can be agreed at scientific advice meetings, which would be viewed the same way during GMP inspections. And of course, there's the complication there that although we have a mutual recognition agreement for inspections, it's an important point that we need to make sure that elements of our GMP approach that we might agree in a meeting with FDA are appropriate during a GMP inspection conducted by a local competent authority in Europe. So if we carry on. And then there's an overarching point which I guess alludes to the slide I used at the start, just to try and highlight the point that we, in the context of prime and breakthrough therapy, what we were looking for is a similar culture from regulators in the context of both review and inspection. So we talk a lot about flexibility and innovative approaches to quality and CMC contents of the file. Are we able to take a similar approach, risk-based, benefit-based, in the context of GMP inspections and ensure that both of those are aligned? And of course, that point I just made about how to make sure we have an aligned global path moving forward is very challenging, but also very important. So if you carry on, I think this is my last slide. So GMP considerations for accelerated access should also consider patient risk and needs in the same way as quality and CMC elements, particularly where there's potential risk to continuity of supply. So the discussion points that we'd like regulators to consider are increased clarity on how GMP considerations for the clinical phase can be appropriate for early commercial supply, harmonised considerations of the risk of GMP inspectional outcomes in the context of what we agree in terms of agreements with regulators at scientific meetings, the use of IMP sites potentially for early commercial supply without requiring full GMP licenses for those sites, and overall how we can make sure that we have an aligned approach to GMP matters as we do for quality and regulatory elements. Sorry, quality and CMC elements during scientific advice type procedures. That was all.
Thank you, Matt. So I think that we can screen the panel discussion. Thanks. So in the panel discussion, I would like, first of all, to invite the other component of the panel discussion, that is Dr. Ramanathan and Dr. Graham from FDA, and Dr. Ganapathy, I hope that I pronounce in correct way, and he has a PhD in analytical chemistry and works at Merck in USA and is affiliated to Third Pharma, and Anthony Lodge from Kite, that it's responsible for Gescart in EU and is working in similar roles in other authorized autologous at TMP at Chiesi GSK over the last 5 years. The questions for the first discussion is about, in particular for the first bullet point with the three sub-bullet point, because the last two will be probably discussed in, but just to take into account, will be discussed in another section of this workshop. The panel is open, and
So I wanted to open it up for my CBER colleagues, first of all, to sort of point out that we do not have a mechanism to release OOS ATMPs at FDA, so that's not a pathway that we have. I'll allow Andrew to speak further to that if he wants to.
We have a different way of handling that flexibility. So we allow investigational use of OOS autologous products, but when it comes to license, our GMP regulations do not allow the release of out-of-spec products. So in that case, we can still allow, if there's no risk to the subjects, investigational use of those products.
Yeah, this is just to clarify the situation for the ATMPs, because I was in the drafting of the GMP guide, and there was a long discussion on this. And in fact, this is not a release process. It's really the administration of a product that is out of specification. So the impact of how this is going to be handled in terms of the pharmacovigilance, for example, is not clear completely. But the fact is that there is no expectation as a medicinal product of this batch, but this batch is administered to fulfill other tasks of the cell-based products that are needed. For example, if you have immune deleted the patient, he has to rebuild his own immune system with part of the product that is not the active part. So there's no expectations on the active substance itself, but on the complex composition that has to be re-administered to the patient. And those are the very exceptional situations that are foreseen. So it's not really to be dealt as a real release of an out of specification.
So I'd like to echo that same sentiment, speaking for small molecules, that it's not really the business to allow release of true OOS material. It's not meeting specification, but there are other pathways within the FDA where we can discuss exceptional release or some kind of regulatory discretion if the public health need really warrants it. I think we really defer to our regulations and our guidance on investigating out of specification results and really identifying if we're talking about laboratory error or some kind of special cause variation. But linking back to what Giampiero talked about earlier on, as far as can you identify the impact to safety and efficacy, and if there really is too much uncertainty where it cannot be discussed or cannot be quantified, release of OOS material really isn't in our framework either. But there are other ways to discuss how can we get product to patients. I do want to make one comment related to the first bullet when it comes to release from IMP sites when it comes to the U.S. system. I think we have a little bit of different flexibility in that since we expect full GMP compliance from phase 2 onwards, full 211 compliance, that it's not really a differentiation between investigational medicinal product sites to commercial sites. It's more of the spectrum and sort of how can you show the bridge and show the comparability between those two facilities or two quality systems when it comes to their oversight and operations. So that sort of mentality and approach makes the next couple questions a little bit easier as far as GMP risk assessment, gap analysis, and then showing that bridging as far as how you can support commercial production or commercial distribution from different types of facilities. So I think that's important for recognition within the group here that in the U.S. system, from phase 2 onwards, we are looking at full 211 compliance.
So just to follow up on sort of so that we do have an OOS guidance out there that talks about, as Mahesh indicated, what to do if you have an OS result. And so it does speak to this rare occurrence where everything is telling you this is not a true OOS, right? But you just can't find the root cause. So you have other testing of the same assay, everything passes, and all the other information on the batch says it's fine, but you can't figure out what the root cause was. There is some flexibility in that region to release the batch at the discretion of the quality system of that firm. But that should be a sort of a rare case, and there is an expectation that there's a very thorough investigation standing behind that decision. In terms of CGMPs for phase 2 onward, as you indicated, we do expect full CGMPs. I think the exception to that is we wouldn't expect process validation to be complete or assay validation to be complete, but otherwise we would expect 210 to 11 to be followed.
I have to slightly disagree with Margarita, not the first time, I reckon. I think for autologous cell products, for which we do not know the dose-response relation very well, and a specification has been set, and there is no time to prepare a new batch for this patient, we can be in a situation where we release it with the objective to treat the patient with the medicinal product with the mode of action as foreseen. So I think that's not limited to different aspects of the product.
I agree. Yes, I'm sorry.
Thank you, Giampiero. Ron Ogilvie from Pfizer. really interesting discussion about OSs. I don't want to get fixated on OSs because, you know, accelerated programs shouldn't be looking for more OSs. So I'll say that with a smile. I'm interested in Scott's comment about the FDA and whether or not products can come from the investigational site. And you heard Scott say, yes, we can find ways to do that. I would just like to make the impolite comment, and I know it's impolite, that if for the patient, the fastest route to get them that commercialized product is to come from an investigational site, and one region can allow for that in their regulatory system, and another region can't, it means the region that can't, their patients won't get that product as quickly. Now, I'll just say that and let that think across the room, really, because that's a concern, isn't it?
Yes, I agree. And for this reason that we discuss about this case study, that probably is an important case study, how to try to find, not now solution, but something that could be discussed in order to make faster and possible the release on a meat product that are really important to arrive on the market quickly and very, because they are for medical need. Perfect. Thank you.
And if I can just permit myself, and thank you, Giampiero, and thank you, Ron, for bringing us back to that key point. I guess the scenario that Giampiero and I have discussed is one where there is no issue with the quality of the product. It is prepared to GMP. The issue, the awkward issue is licensing. There's a legal barrier, but not a quality barrier in the context of what we think is appropriate for supply. And I think, Giampiero, it's fair to say that we've been talking about this is a problem we need to address, and we're not going to solve that today. But I think what we can potentially, I'm looking around the room today, agree is that actually in the context of what is appropriate to patient safety and the health risk benefit, it is appropriate to do it. The issue is a legislative one.
I agree. There was a colleague. Yes.
Yes, I'm Therese Saunders from the Norwegian Medicines Agency. I can assure you that we are dealing with these issues right now. We have products on the market. I'm not going to mention any names, but we are really dealing with it. And then, you know, the product needs, especially with the autologous products, the patient needs the products back. And if it's out of spec, then how to deal with that? But we now have a procedure, actually, that we have more or less accepted, at least from the EMA. But the problem is that we have national legislations in Europe. So all the countries kind of decides how to proceed with this themselves. And that's the problem. In the US, it's more easy because then it's the FDA and it's just one authority. But in Europe, it's several. But we are trying to deal with it, and we are trying to give the patient, in case of cells, as I mean, in most of these cases, I mean, even though it's an out of spec, whatever, it can be color, it can be viability, whatever. But in most cases, I mean, the patient doesn't have any other opportunity. So he will get his cells back.
Thank you. I've seen one hand raised there.
Yes, Mairead Duke from Biomaran. Matt, can you help me understand, in the filing for this approach, what have you described in your drug substance or drug product manufacturing section to facilitate or to ask for this?
So it's a hybrid case study that has occurred many times. I'm saying that we haven't done it. The point we were trying to raise is that it's not possible, but there's no reason for it. That was before we knew that AZ had done it. So maybe Stuart, sorry, can answer the question.
I'm sorry, Stuart, before you. There was Margarita that was asked to add something. Sorry.
Just very briefly, the GMP guide, and contradicting what Marcel was saying, it is really on the administration of the cell components, because the cells, it is only applicable to the cells and only in that exceptional circumstances. Is not open, for example, if you have reduced potency or if you have an out of spec of characterization, other specifications. It's really on the need to release the cells that are, and it's only that particular case, and it's out of the release program somehow. So we have to really be careful not to think that out of specifications can be really generally considered, because that's not the situation. That it's only, and it is exceptionally, the administration of the cells and tissues that are contained in a cell tissue-based ATMP that is out of specification may be necessary for the patient. And that is only the scenario. It is well written down in the GMP guide that is only on that scenario. So we have to really be careful on the discussion of releasing out of spec, because that is not the situation.
Thank you, Margarita, for the clarification. So there was Stuart, thanks.
So to answer Marie's point, I think, you know, the two things were crucial. The fact that we could demonstrate that we were using the same PQS across both the clinical and the commercial site to all intents and purposes, and also the volume of information that we could supply to show good compatibility and good control over the process throughout the clinical and the anticipated commercial. So those two things, as well as Mahesh has said, really, really active engagement during review, which, you know, it's not really a data thing, but I can't help but think that that was an underpinning of the success there, is that engagement.
I agree. Yes.
Hello there, Anthony Lodge from Kite. So I think coming back to the out of specification question for ATMPs, I think, you know, one question we have is when does exceptional no longer become exceptional? We're about to launch in Europe. We already have clinical and commercial experience in the US as well as Europe. We know that our specifications for the commercial product are tighter than what we set in clinical, so we're expecting some out of specification batches. It will be a low percentage, around about 3% based on experience. So in Europe, we're going to use the GMP guideline to be able to release those as out of specification products. Obviously, in the US, we've got a different pathway, but I think, you know, based on continuing manufacturing experience, understanding batch data on an ongoing basis. The problem with the out of specification products that we're releasing or expecting to release is that we can trace that out of specification back to the quality of the apheresis starting material. The patients themselves are very sick. They will have undergone a number of different treatments already, which will have affected their cells and the ability to be transduced, for example. So we understand the reasons why out of specification may arise. So going back to, you know, the discussion around continuing manufacturing experience, is it possible that an exceptional circumstance initially will become the norm?
I'm sorry, go ahead. Sorry, go ahead after you.
Sorry.
My one last comment on the inspections is, while we understand that the European agencies have they do certify the clinical manufacturing sites. It's not typically done by the US authorities. Where I think we need to probably look forward to is, from an industry perspective, is the expectations that one has to see if we use an IMP site for commercial manufacturing, pre-approval inspection expectations from FDA and EMA or EU, would that there may be discrepancies in the expectations, and that needs to be a critical factor in how industry can actually begin to see, can we use the IMP sites.
I think in the context of the OS discussion, I'm convinced that we need to slightly rethink our way we set specifications for our autologous products. We are partially in a situation of OS because we were setting our specifications the traditional way, looking at process performance, and manufacturing history, and not sufficiently taking consideration our understanding of correlational analysis to clinical outcome. So really weighing what is the importance of a certain attribute. And at the same time also, and this is where the learning comes in, underestimating the process variability that you're looking at when you globally expand to other sites, when you change your workforce from a very scientific one that you have in the beginning in cell manufacturing to a more industrialized workforce. This comes with a lot more variability than we ever expected. And I think this is a learning curve we're in right now. And we are very much appreciating working with the agencies on establishing how we can give the patients access to their product. Most of the cases we can show very clearly due to our correlation analysis that there is no risk to the patient by receiving that product. And I would be very hesitant to put additional hurdles up to give rapid access to that patient who is basically having no other treatment options.
Michael and Shalexion, just to add to that, as Ron and it might have been Andrew said, we don't understand what attributes are important yet. The learning curve is so skewed towards post-approval because we're doing things more rapidly and quicker earlier to get first to market. And it goes to the comment that we may need to think about innovative approaches. It's not quite a round hole square peg, but we are trying to fit in things like knowledge, control strategy, which have been around a long time, into something that is vastly different in terms of the amount of data we have at the time. So.
Just to add one thing about the comment that was raised about the GMP compliance. So in Europe, we have GMP approval for IMP and for commercial products. So it means that probably this could facilitate an approach to move forward from the IMP to commercial in order to release product from the IMP, because obviously considering that they were assessed by an inspection and were approved and authorized, it means that the pharmaceutical quality system was already evaluated, obviously according to the IMP, not according to the commercial manufacturing, that it means a different approach for validation and so on, because it's an increasing system during the development. But this could help, and just to understand how to which tools we can find and to discuss about it.
I think we need last comment, please, because we're going late.
Just 10 seconds. Giampiero, the comment that you made, I fully agree with that. But I think, it goes back to what Ron said about the global acceptance of that. It may be, very different between different countries.
Please, Mahesh.
So staying on the IMP discussion, just to reiterate and to support what Giampiero is saying here is that even though it can be done in the U.S. system, we don't do it lightly, right? We want to make sure that the PQS of the site is available for inspection, or we have inspected it. It meets our commercial expectations. But we encounter these scenarios, and we understand that launch may be critical for a short-term solution to get supply to the patient, but we are also interested in the durability of supply. Again, we're talking about access to care here, and it's not just the fastest way and that's it. We want to know, how are you going to maintain this over time? So when we talk about existing tools or tools that can be used better to have that conversation, post-approval change management protocols or plans, or even life cycle control approaches to help us understand how are you going to move from site to site and how are you going to increase the durability of supply, really help these proposals work.